Hi there, Mark here again. In this video, we're going to be doing the third step in our paper modeling activity, and this is going to be where we deal with the actual genetic payload of a coronavirus. In other words, what's that RNA strand on the inside doing? How can we model it with paper, and what happens to it as it gets into the host cell? Like the other paper activities in this series of web resources, this one is available as color, as black and white, and as a drawing template in case you have no printer at all. And again, just like the other ones, you'll start by cutting out all the components in the layout, following the dashed or dotted lines. And I'm going to speed this up because there's no reason to watch me cut paper for 20 minutes. So what did we just cut out? What are you left with? Well, first you have right here, this is the viral genome. So these are the genes, the RNA strand that's inside our virus. And in fact, if you want to, you can kind of coil this up and fit it right inside of our coronavirus model. That's where it would be housed. It'd be held inside this envelope of our coronavirus. And only after attaching to the host cell would it be released and put into the host cell. There's this extra little component right here. It's kind of an optional thing to print. Essentially, it's so that you can show any of this area on this long graphic here is really nothing more than RNA sequence. So A, U, G, and C, nucleotide sequence. That's all that this graphic is supposed to let you show. What happens once this is now inside the host cell? Well, it's an RNA genome, and so it begins to be translated right away by a ribosome into a protein. And in this case, it's actually not just a protein, it's what's called a polyprotein. We'll be talking about this in more detail in another video, but it's not just one protein, it's a variety of proteins. That's what this second piece of paper, this second strip represents. And what happens is this polyprotein then needs to be cut into all the individual proteins that will then form new viruses. You'll notice that the very first protein in this long polyprotein strand that we've translated from our viral genome is called a protease. A protease is a specific type of protein that is able to cut protein strands. So this starts actually by cutting itself from the rest of the polyprotein. So you now have a protease floating around free, and it goes and further cuts the rest of this polyprotein. Everywhere that there's a protease cut site, it will cut another one of these polyproteins into its own individual piece. And we can simulate that simply by cutting each one of these with our scissors. So in the end, what you're left with is a viral genome that was inserted into the host cell. That was translated into a long polyprotein with a number of different proteins all connected. And then our key protease protein went back and cut each of those individual proteins into their own individual pieces. Alright, so by the end of this next step in this activity, you should have your coronavirus, you should have some spike proteins that allow it to dock onto the ACE2 receptor on your host cell membrane, and now we've injected our genetic payload, our strand of RNA, into the host cell. And we've not only inserted it into our host cell, but we've translated it into a long polyprotein, and we've cut it into those individual proteins that are needed to create more viruses using that key protease protein.